Hello friends, Ace Botany, next episode, Sexual Reproduction in Flowering Plants, 6th one. One more we need in this chapter. And what all things we will be discussing in this chapter. So I will show it by sharing the screen. I think the screen came. Under post-fertilization events, episode 6. Endosperm development we discussed in the last episode. In the 6th episode, present one, what we are discussing. Here two aspects, embryo development and ovules into seed. Fourth item, ovary into fruit, etc. Apomixis, all that area we'll be discussing in next episode. So we can start with the embryo development. Embryophyte starts in plant kingdom from bryophyte. Bryophyte, pteridophyte, gymnosperm, angiosperm, embryophyte. And ploidy of the embryo is diploid. And zygote develops into embryo. See, for the development of embryo, food is a must. So after formation of some endosperm only, zygote develops into embryo and continues its procedures. Otherwise, no nutrition. And embryogeny in dicot and monocots have some similarities in the initial steps up to globular stage. Then slight deviation. I think first 5 statements clear. Now we have to proceed with the dicot embryogenesis. This 6 diagram, purely NCERT labeling question. But from that, you may not be able to get much idea. So what I have done, I have drawn a slide to explain this dicot embryogenesis and monocot embryogenesis. So that I will explain. Now this is how it starts. See, first, zygote. Zygote is otherwise called oospore. Oospore can be called as fertilized oosphere. Oospore is zygote, it is diploid. Oosphere is haploid, that is equivalent to egg. Fertilized oosphere is oospore. I think the 3 terms clear and they are ploidy level. So zygote undergo a division. 2 unequal cells will be formed. And large cell, it is called suspensor cell towards micropylar area. Small cell, which is called embryo cell, towards the Chalas cell area. And suspensor cell, which undergo transverse division to form 6 to 10 cells. So number is 6 to 10. And tip cell of the suspensor, which swells. And that cell is called vesicle cell or ostorial cell. And the bottom cell of the suspensor is called hypophysis, which develops into radical tip. That is regarding suspensor cell fate. Now coming into embryonal cell, which divides vertically twice and transversely once to form 2-tiered 8-celled embryo. So embryonal cell vertically divides twice, transversely once to form 2-tiered 8-celled embryo. In it, epibasal tier gives rise to 2 cotyledons and plumule. Hypobasal tier gives rise to hypocotyle and majority of the radicals. Now, this octant embryos undergo periclinal divisions and different areas will be formed, including ground meristem, procambium, dermatogen, radicle, etc. Again, division proceeds. It grows, reaches into heart-shaped embryo. Later, it becomes mature embryo with 2 cotyledons. That is how the dicot embryogeny takes place. And here you can find a diagram, that is NCERT diagram, typical dicot embryo. I think part is clear. Now we can proceed to next area. Yes. Now when we analyze part of a typical embryo, three areas we can find: epicotyle, hypocotyle, embryonal axis. These are the three areas: epicotyle, hypocotyle, embryonal axis. Here you can find B is missing. Embryonal axis. Okay. So embryonal axis, otherwise called tigellum. Epicotyle, hypocotyle, embryonal axis. Now what is epicotyle? Epicotyle is connected with the stem, and that is the portion of the embryonal axis which is above the level of cotyledon, which ends into tip of the stem or plumule. So epicotyle connected with the stem, portion of the embryonal axis which is above the level of cotyledon, which ends into tip of the stem or plumule. Hypocotyle, portion below the level of cotyledons, which ends into root tip or radicle. So hypocotyle connected with the root part, epicotyle connected with the stem part. Root tip is very delicate, so it has to be protected. That is done by a structure called root cap or calyptra. And the last part is embryonal axis, which is otherwise called tigellum. So three area clear, I think. And in embryogeny, there is an order. Starts with the zygote, followed by proembryo, then globular stage, heart shape, and mature embryo. Now we can find two NCERT diagrams. Both are important for theory as well as practicals. NEET exam, labeling question, to draw questions. Monocot embryo, when we check, we can find total 4 star will be marked there. One is scutellum. What is scutellum? Single cotyledon in monocot is called scutellum. And what is epiblast? Remnants of the second cotyledon in monocot. It is called epiblast. Remnants of the second cotyledon in monocot. It is called epiblast. It is a labeling question, theory question, entrance question. What is meant by epiblast? Is it seen in dicot or monocot? What it represents? Epiblast is the remnants of the second cotyledon in monocot. I think that page is clear. Now we can proceed to monocot embryogeny. In the monocot embryogeny, zygote has got a transverse division, suspensor cell and embryonal cell. Suspensor cell towards micropyle, embryo cell towards cella cell. Embryo cell divides transversely, top cell and middle cell. Then terminal cell divides vertically and transversely to form embryo. And here you can find NCERT diagram, this one, with the scutellum and epiblast. Epiblast is the suppressed second cotyledon in monocots. I think that page is clear. Now, next aspect for us is seed. What is a seed? A seed is a fertilized ovule. And study of seed is called spermology. Because in plant kingdom, we study two groups of plants, gymnosperm and angiosperm, with the suffix sperm. Sperm means seed. So automatically, it is another question plant kingdom, spermatophyta starts with, ends with. Seed we can only find in gymnosperm and angiosperm. So gymnosperm and angiosperm can be otherwise called spermatophyta, seed-bearing plants. Number of seeds variable. Now various classification for seeds. Albuminous or endospermic seed, that is remaining. Last episode we discussed it. Exalbuminous or non-endospermic seed. Endosperm will be used early. Now another term, perispermic seeds. What is perisperm? Perisperm means remnants of nucellus that remains in the seed. And typical example for perispermic seed, famous question NCERT, black pepper and beet. And ploidy of the nucellus is diploid. Now we have to proceed with the structure of seeds. a seed has got three specific areas. One is seed coat, one is endosperm, one is embryo. Seed coat for protection, endosperm for food, embryo that is develops into plant. Now seed coat. From where the seed coat develops. In female part when we studied, you may be remembering indegments, outer and inner indegments. And these two indegments converts into seed coat. Outer one convert into testa, inner one convert into tegment. If both these two indegments are there, converting into seed coat, they are called bitegmic. If only single covering, it is called unitegmic. If it is absent, it is ategmic. Testa will be outer, tegment will be inner. And here also there is a small opening, it is called micropyle. And what is the aim? So micropyle is a small opening in seed coat which facilitate the entry of water as well as oxygen in connection with the seed germination. So micropyle is a small opening in seed coat which facilitate the entry of water and oxygen for the seed to germinate. Now you can find the second diagram, that is maize grain, a typical maize grain monocot where everything is marked. It is an important question for diagram based labeling. Aileron layer around endosperm only. Endosperm is triploid. Aileron layer also triploid. Then you can find castor. Then onion is shown. It is a labeling question where people will not look onto it. It is NCERT diagram. I think that part is clear. Now comes next area, that is called seed dormancy and quiescence. Dormancy and quiescence used as synonym, but slight difference is there. Dormancy is more towards endogenous control. Quiescence is more towards exogenous control. Now, dormancy, is it good or bad? That we can decide only depending on the situation, environment. If suitable conditions are available, a dormant seed should germinate. Otherwise, better suppress. If moisture content comes to 10 to 15%, that can lead to dormancy. Many factors can lead to dormancy, specifically adversity. One is moisture content. If it is 10 to 15%, seed will become dormant. Now comes germination. Germination means from seed, seedlings will be developing. It is called germination. And what is the reason or why nature prefers seeds? Seeds has got many advantages. They can survive certain extreme conditions because they got protection and they got food reserves. Second, seeds are formed after sexual reproduction, so new combinations will be there, genetic combinations. So that will help to face adversity. And seeds also involved in agriculture because it can be stored for a long time, no crops can be raised. Now comes viability of seeds. Viability is present in many areas. Pollen viability, seed viability. And in pollen viability or seed viability test will be common type. It is based on oxidation reduction. So the famous test for viability, it is called TTC test, triphenyl tetrazolium chloride test. It is not in syllabus. A few years back, they asked which is the test for seed viability. TTC test. Same test can be applicable in various other areas, including pollen viability. Longest viable seed, NCERT 2 given, 2 items given. One is lupin, Lupinus articus, Arctic tundra, 10,000 years of dormancy. Then date palm, Phoenix dactylifera, concerned with the King Herod's palace, 2,000 years old. These two are the longest viable seeds, keeping dormancy for a long time, then it is able to germinate. How we can break seed dormancy? One is called scarification, thinning of seed coat. Other is called stratification, temperature treatment. And identifying the inhibitors such as ABA, coumaric acid, phenolic derivatives, and suppress it by using chemicals against it. And fourth is treating with the gibberellic acid, which helps to break seed dormancy. So these are the main topics what we have to discuss in this episode. After that, as usual, 5 theory question, 5 NEET model question with the key. So what all things we studied today? 6th episode of sexual reproduction in flowering plants. In this episode, we are mainly dealing with the embryo development and ovules into seed. One more episode needed to finish this chapter. Then we started with embryo development, embryophyta, ploidy, zygote into embryo, and why that is endosperm has to develop first before zygote develops into embryo. Then embryogeny in dicot and monocot, similar up to globular stage. Then dicot embryogenesis, division from the zygote, unequal cells, suspensor cell, embryo cell, posterior or vesicle cell, hypophysis. Then embryonal cell vertically divides twice and transversely once to form two-tiered 8 cells. Then epibasal tier, 2 cotyledonal plumule, hypobasal tier, hypocotyle and majority of radicle. Division continues. Different areas and layers will be formed like ground meristem, dermatogen, procambium, radicle. Then it enters into heart shape, finally into mature embryo with the 2 cotyledons. Then parts of a typical embryo, epicotyle, hypocotyle, tigellum or embryonal axis. Then order of the embryogeny, zygote, proembryo, globular shape, heart shape, mature embryo. Then 2 diagrams, dicot embryo and monocot embryo. Monocot embryo specifically scutellum, single cotyledon monocot, epiblast, remnants of the suppressed cotyledon in monocot. Labeling question. Then monocot embryogeny. Then we discussed entered into seed. What is seed? Study of seed, number of seed, types of seed, perispermic seed with example black pepper and beet, structure of seed, seed coat, endosperm, embryo, testa, tegment, bitegmic, unit tegmic, hetegmic, endosperm, ploidy, triploid, function, nutrition. Then embryo, diploid. Then 4 diagrams including maize grain and onion. Now comes seed dormancy. What is dormancy? How it differs from quiescence? Is it good or bad? And what is the percentage of moisture needed? Otherwise, it will be undergoing dormant stage, germination. Then, what is the advantage of seeds in agriculture? Protection, food availability, then new combinations. Then, it can be stored, seeds can be stored. Seed viability test, TTC test. Viability test is applicable in various areas, mainly oxidation reduction reaction. Problem viability is the same test, triphenyl tetrasodium chloride test. Then 2 important NCERT data. Longest viable seed after dormancy, that is lupin, 10,000 years of dormancy. Second is date palm, Phoenix dactylifera, King Herod's Palace, 2,000-year-old. Now how to break seed dormancy, scarification, stratification, nullifying the inhibitor, treating with the gibberellic acid. Then as usual, 5 theory question, 5 entrance question, and we'll be continuing one more episode to finish this chapter, last part. And if you are like this episode, subscribe or like it, share it, and put comments. We can meet later with last episode of sexual reproduction flowering plants. Thank you.